Okay, so I'm going to go over protein synthesis, which includes transcription and translation. And so first I'm going to start out with just kind of going over DNA and the relationship between genes and chromosomes and DNA. And so we know that DNA starts out in the nucleus of a cell. And so we have the nucleus here, and it starts out as just, very uncondensed, a substance called chromatin. So it starts out as chromatin. And then when the cell starts to divide, that chromatin condenses into a chromosome. And so we can kind of look at chromosomes like this. And each chromosome is made up of strands of DNA. And we know that we have, and DNA is double-stranded, right? And we know that we have 23 chromosomes. Humans have 23 chromosomes. And then in each section of the DNA, we can see genes for different proteins. Genes code for proteins. And so, this might be the gene for hemoglobin, depending on what chromosome it is. So each chromosome contains genes for different proteins. And so genes are basically just sections of a chromosome. And so when a cell, or when you need a certain protein, that section of the DNA in the chromosome unwinds, and that chromosome, or that section, that DNA that gene contains, is transcribed and then translated into a protein. So there's two processes that occur in order to make this protein. The first process is transcription, and transcription occurs in the nucleus because the DNA can never leave the nucleus. Okay, so all the instructions for the proteins are in the nucleus, in the chromosomes, in the nucleus. And those instructions are very important. So they never leave the nucleus, because if they leave the nucleus, something could happen. It could be damaged because they have to go on quite a journey to get to the ribosome where translation occurs. So basically what happens is we make a copy of that gene in the nucleus, and then that copy is taken to the ribosome. And at the ribosome, we have the process of translation. Oops, not ribosome, it's ribosome. Okay, so transcription occurs in the nucleus, translation occurs at the ribosome. And so we'll first go over transcription, and then we'll go over translation. Okay, so let's say that we have a section of chromosome, or a section of the DNA in a chromosome that has unwound, right? And we have these different nucleotide bases. And then we have the complementary bases on the other strand. Oops, that's wrong, because A or G pairs with C, and then G, G, C, T, A. So these are called complementary base pairs, right? In DNA, A always pairs with T, and G always pairs with C. Okay? So one of these is going to be our template strand. One of them is the coding strand. So we're going to say that our this bottom blue strand is our template strand, and the top strand is our coding strand. So this strand is the code for the gene. This strand is the template strand because RNA is going to make a copy or a complementary copy of the template strand code. All right. So our first step in protein synthesis is transcription. So we are going to transcribe the template strand using RNA. And remember, RNA uses U instead of A. And so, U instead of T. And so in RNA, A always pairs with U and G always pairs with C. So U replaces the T. So if we go through and we make a copy or transcribe this template strand, our template is going to be, let's see, so this would be A, U, A, G, C, C, G, A, U. So this is our RNA. This is our RNA transcript. So our RNA transcript, AUA, G, C, C, G, A, U. And we're going to put another one on the end here. We're going to put UAG on the end, just so we have we have 12. Just so we have 12 nucleotides in our transcript. So this is called our pre-mRNA. mRNA, remember, stands for messenger RNA. That's the one that transcribes that message, those instructions for the gene. In order to prepare the pre-mRNA to leave the nucleus, we first have to take out the introns, and splice together the exons, and then add a poly A tail and a G cap. We would add a poly A tail to the 3' end and a 5' G cap. But for our purposes, we are going to just, we're not going to do the RNA processing. We're just going to take this as our final mRNA, or our edited mRNA. I have another video that I'll send you that has the editing part. So, but we're just going to take this as our edited, our final RNA. Okay. And so we can now, this is ready to leave the nucleus. It's going to leave the nucleus, and it's going to travel to the ribosome for translation. All right. And so we're going to take that strand, and I'm going to try to remember what that was. So let's say it's AUG. Let's just pretend that this is. So it's AUG, C, C, A, U, G, G, and C, A, C. Okay. So this is our transcript. Okay, so this is our transcript. And so now the ribosome is going to come along and it's going to read this code in a series of 3 letters at a time. Each of these are called codons. And these codons are going to code for amino acids. Okay, so we have to use a codon chart in order to decode this transcript. And the codon chart can look something like this. And so if this is our, so let's say this is our transcript, AUG, CCA, GU, UAG. So again, we're going to read this. The ribosome is going to read this in a series of 3-letter codes. And remember, these are our codons. All right? And so if we look at the first one is usually going to be methionine. AUG always codes for methionine. And we can look on here, AUG, so we start here, A, and then U, and then G, right? So our first amino acid is going to be MET, which stands for methionine. Our second amino acid is CCA, so we look at CCA, and that's proline, PRO. Our third amino acid is GUA, so it's G, U, and then A, which is, we need to go back, which is valine, VAL. And then our last one is UAG, and UAG codes for stop, and so we just put our stop there. And that means that the ribosome can disengage and we're done producing that protein. And so our protein consists of these amino acids. Now, in reality, proteins are much longer than 3 amino acids. But for our purpose, that's what we're going to use. So I hope this helped. If you have any questions, let me know.